Hey all, welcome to Chapter 8, Round 1 of Chapter 8 notes. I'll apologize in advance because the whole family is at home today, so it might be a little noisy in the background. So this chapter is focuses on cell reproduction, and then we'll kind of segue into how cell reproduction lays the foundation for inheritance. All right. So before we get rolling, just have a look at the first question here and pause the video and see if you can come up with what purpose does cell division serve? All right, pause and then you can restart for some of the ideas there. Okay, so if you're thinking along the lines, cell division occurs for a number of different reasons. One is for growth of the organism, right? Growth of parts of the organism, organs, and tissues. It also occurs, cells in our body die constantly and need to be replaced. So you're losing about, I don't know, 50 million skin cells a day. So when you sweep up those little dust bunnies in the corner, there's probably a few million dead skin cells in those little dust bunnies, or replacing damaged cells, cells that are injured. And over here in blue, I've written propagation of the species. So cell reproduction occurs for the continuation of the species, much like when we produce sperm and egg to produce more offspring. There are essentially two types, and each of these can be categorized as one of two types of cell division. That's mitosis. So these ones over in red fall under the category of mitosis. And in the case of sexual reproduction or propagation of the species, uses the form of cell division called meiosis. They're very similar. We'll look at the differences between the two. So if you look at, so we'll start with mitosis first. And if you look at mitosis, it serves different purposes in different organisms. So if you're a simple bacteria, then your method of propagation of the species, continuation of the species, is cell division by, and maybe I should have done this in red since we're talking about mitosis, but this binary fission in which all bacteria, including the E. coli living in your intestines, this cell division occurs in all bacteria, and it's basically binary fission is just a form of mitosis. These are all these examples that I'm showing here are forms of mitosis, right? So mitosis includes reproduction in single-celled organisms. It includes dividing in animal cells, like any cell in our body divides via mitosis. Whether you're making new cells as the organ gets bigger or replacing cells that die, or in some organisms, the process of regeneration is also mitosis. And in some organisms, that can be quite extensive. If you want to check out a very cool video, check out the axolotls, which are native to, I think, lower North America, South America, kind of a salamander-ish looking organism, and they have unbelievable regeneration abilities. Literally, you could sever an entire limb. It is nervous tissue, bone tissue, muscle tissue, and they could regenerate that entire limb. In fact, there's even weird scientific research going on where they're, it sounds bizarre, but growing literally another head on the same organism. Check it out; it's pretty insane stuff. And of course, research in that field would be looking at our own regeneration of nerve cells for spinal cord injuries and that kind of stuff. We do a certain amount of regeneration, of course, right? You get cut a little chunk off the end of your finger, it's going to regenerate relatively smoothly. You can donate part of your liver, right, to somebody, and you would regenerate that chunk of liver that you've donated. And then, of course, our complete early human development, right? So, as I said up above, we're formed from sperm and egg, right? Sperm and egg, we get fertilization, and fertilization produces a cell, which we call the first cell that we all start off as, is called a zygote. And this zygote will divide into two cells, which divides into 4 and 8, and so on. And you've got an embryo developing, And eventually you go from a single cell to an individual. And I should have done this in red as well, but I wanted to emphasize this part is the sexual reproduction. But then from here on out, zygote to individual is all through mitosis. All right. And mitosis, basically, in the most simplest description, is just copying of the genetic material, right? So when we think of a cell, right, and inside that cell we've got a nucleus with a bunch of DNA, right? Well, this DNA, right, has to be copied before the cell can divide. So DNA must be copied, and copies shared between the two new cells. All right, and it's this it's the sharing of that DNA, that copied DNA, that represents mitosis, right? And so uh, that's what we're going to look at in detail down below. And just before we do, I just want to go through the terminology that we use around the molecule DNA. So we've introduced DNA from chapter 4. We know that it's our hereditary material. We know it's a nucleic acid, and it comes in many forms in the cell depending on what's going on in the cell. So we've got all of these different terms over here represent DNA in different forms. So the unraveled straight up double helix, right, is DNA. And this is the form DNA would be in when the cell is making proteins, right? You have to be able to access the genetic code. So making proteins need access to the genetic code. DNA exists in that double helix. And I should say that DNA is always in the double helix, even as it goes through all of these different forms, right? But here it's essentially unraveled, right? So you have to access the genetic code. All right, and in order for, it's maybe hard to imagine, but we have about 6 feet of DNA in every single cell in your body. It's a bit insane. And imagine that it's got to fit into this microscopic structure that is our nucleus. And so the cell has a very effective way. Kind of think about it when you try and stuff your clothes into the drawer, right? Well, you're maybe not going to get as many, or if you think about stuffing them into your suitcase the last time you took a trip, maybe a year and a half ago. But if you pack in a very organized way, you're going to fit a lot more stuff in there, right? And that's essentially the process of packing DNA into the nucleus as well. It wraps around these molecules called histones, and those histones join together with the DNA wrapped around, forming what you know, and some of you brought up in your chapter 4 assignment or presentation, a chromatin fiber, right? And those chromatin fibers will continue to wind and wind and wind, wrapping around even further, making a structure called a chromosome. And pretty sure we're all familiar with that. And this is the form DNA takes when cells get ready to divide. Form DNA takes for cell division. All right. And the picture and the image down below just kind of shows all those different forms, both the sort of artistic impression over on this side, and as well the electron micrographs, right, of what it would actually look like in a real cell. And I just want you to sort of pay attention, and not that I would ever ask you about any of that, but just you can see how the size of it differs, right? So the straight up double helix is just 2 nanometers across, but as you build in these histones and it wraps around there, it becomes a little bit thicker, and then as those histones wrap and form those double layers there, you get an even thicker strand all the way up to the chromosome here down at the bottom, and you can see the electron micrograph quite a bit thicker if we look from end to end, 1400 nanometers. So almost 700 times greater in size than the individual helix itself. And so this is visible through many light microscopes, maybe not quite to the detail you're seeing there, but certainly you can see chromosomes under a regular light microscope that you would have used in high school or what we have in our lab at the college. One thing I want to point out is this characteristic shape. Typically, a chromosome looks like this. So this is a typical chromosome, and when it looks, we often see them represented as these kind of X-shaped molecules, and this is what a chromosome looks like in its duplicated form. So this is a duplicated, that's when the DNA has been copied, duplicated form of chromosome. Okay, and part of the reason why they have this X-like shape is in the middle here, in this section, is a protein area that is called a centromere. And this centromere, right, that we see in this indentation, right, these centromeres play a role in the cell division of moving DNA around, right? Play a role in cell division to move chromosomes. And we'll look at that a little bit more. You'll look at that when you do some notes. You guys are going to do some of your own notes, and I'll point that out in a second. All right, so that's just a little bit about sort of DNA terminology, and we'll use all these terms at different times. So the next thing in terms of understanding the process of cell reproduction is really we want to understand the concept of the cell cycle. And the cell cycle isn't anything different than when you think of your own life cycle. You know, you're born, you grow, and as you grow, you carry out your life functions, whatever those might be, right? And at some point in time, maybe you might reproduce, and maybe you won't, right? And that's true of lots of cells in your body as well. Some cells will reach a point of reproduction, and probably in a much shorter time period, of course, than it takes us. And some other cells won't. They'll enter into a certain phase in which they just are doing their business and that's it. But most of our cells in our body enter a stage where they reproduce. And then once they've reproduced, they'll continue going on. They'll grow again and continue carrying out their functions until such time that they die, right? Just like us. And that process, that cell death, is referred to as apoptosis. But in a nutshell, that represents the life cycle of a cell, right? A cell is produced, it grows, it carries out its functions, and then apoptosis takes place. Okay, so the cell cycle is basically divided into these three main stages shown here on the side. And I would like you to read this section in your textbook, all right? Just have a read through page 129, all right? Page 129. And just think about those different stages and then restart the video to get the details in the descriptions. Okay, so these stages of the cell cycle broken down into interphase, mitosis, and cytokinesis. So in black, I've tried to indicate over here the stages of interphase itself, which represents the majority of the life cycle of the cell. And it's divided into three stages as well, one G1, S phase, and G2. And we'll look at these in more detail in the next video. But essentially, each of these different stages of interphase include any one of the following things going on here. So the G phases are emphasized by growth, right? So this would include particularly G1, where the cell grows. It's going to produce more phospholipids to build that bilayer bigger. It's going to manufacture more organelles, and specifically, it's going to be carrying out whatever functions that cell is designated to do. If it's a liver cell, detoxifying the blood, producing various hormones, or manufacturing bile, which we'll see in the digestive system, but it's carrying out what it's supposed to do. If it's in the S phase, this is the actual phase of duplicating DNA. So the S is kind of like synthesis. And when it re-enters back into a G2 phase, that's another phase of growth, but here it's focused on manufacturing proteins and stuff, getting ready for cell division, right? But all of these collectively together represent interphase, upon which at the end of G2, the cell is then prepared. It's done all the stuff it needs to do in G2. It's prepared to go through mitosis. And that's what I've indicated down here in red and in blue. This is the process of dividing the nuclei and the genetic material, which goes through 4 steps: prophase, metaphase, anaphase, and telophase. And you guys are going to take your own notes on this. I'll explain that in just a second. But at the end of that is cytokinesis, which is basically the division of the cell contents. All right, division of cell contents, and literally the using of these microfilaments. So here we get another look at the components of the cytoskeleton, and specifically microfilaments that work just like that drawstring, maybe on your gym bag or on a small bag, where you just pull that drawstring and it kind of pinches the top off. Well, those microfilaments act in the same way, pinching the cell in half. But prior to doing so, there's kind of this division of cell contents being organelles and even the cytoplasm itself. You know, ensuring that each cell kind of has a starting point of specific organelles, some cytoplasm, right, some cellular fluid, etc., and then the cell pinches in half. All right, that's the end of the video. So what I want you guys to do for your own notes is I want you to go through and do the details of prophase, metaphase, anaphase, and telophase. So I want you to use page 130 and 131 and just include a diagram. All right, and use the little cartoon diagram that's shown if you have your book open. Make sure it's labeled, because you need to know those specific structures. And then, you know, 2 to 3 point form details about what's going on in each stage. 2 to 3 point form details for each stage. So try to have a read through the paragraph and, you know, just summarize the main key points. All right, that's video number 1. I'll be posting that and video number 2 shortly.